Alright, gang. Lecture 5. The war is almost over. At least for now. Yeah, so I think what I want to talk about here is going to be a couple of things. So they're gonna, Professor Kong talked a little bit about more about translocation and transmembrane proteins, and then we're also going to talk about this. This is the main part of this, the post-translational modifications. I'm not going to harp on this too much. It's important, sure, but I think we covered a lot of this already. So, already talked about this. So, some of the quite people were asking, like, why is the N-terminal end in the ER lumen and not the cytosol? It's because, remember, I showed this in the previous video. When you initially put the protein in, first of all, there's no positive charges at all that denote the sidedness. So, it's just going to be based off how it enters the ER in the first place. So remember, there actually should be basically another section here like this. This should be the end terminus right here, but that gets cleaved. So basically this flops down like that, and so basically just does this. It doesn't make sense for it to have gone in like, I can't even draw it, but it doesn't even make sense for it to have gone in like this. Because remember, most of the time when these proteins are actually put in, they're naturally going to be put in this form, right? As you remember from the images. And then when you cleave it, it falls down. But naturally, they are put in this form, unless there is charge somewhere. If there's charge somewhere, that changes, but yeah. So, making soluble membrane-bound proteins. Talked about this already. I'm not going to bother about it as much, so yeah. If you want to take a look at more of these in detail, feel free to watch Professor Kong's video, the main video. That'll be a useful refresher on these. Or go back and watch my video if you want to use that. Yeah. What about if the star transfer again, talked about these already, not going to worry about this. Again, yeah, this we treat it the same as internal signal sequence, yeah. So, yeah, again, I think these are all really well explained already by, or at least they were discussed a little bit in my previous video, and again, Professor Kong does a great job of kind of going over all of these to make sure that you fully understand it, so I'm not going to harp on it. For this, actually, what happens if there's only one signal sequence at the C-terminal end? Let me... I'm actually not too sure in this case. I believe it would still get stuck in the membrane or the Golgi. But if it's like at the very, very end, then it doesn't get stuck in the Golgi. Or sorry, not Golgi. If it's at the very, very end of the protein, it doesn't act as a start sequence. It just, the protein's basically fed into the ER normally, right? It's not, and then it's like cleaved and it's like released into the ER. It doesn't act as a transmembrane protein in that case, because remember that transmembrane kind of, those transmembrane features only really show up if the signal sequences are in the beginning at the N terminus and in the middle of the protein. Not if it's at the end of the protein. So it has to be in the beginning or the middle. Even if it's in the beginning, usually actually it doesn't really end up as a transmembrane protein. If there's a signal sequence in the beginning, there also has to be a signal sequence in the middle for it to be a transmembrane protein. So if it's all the way at the C terminus, the protein just gets translated like normal. It may not even go into the ER, right? Because, you know, there needs to be a hydrophobic region to signify going to the ER, and if this thing is just at the end of the protein, it's never going to get the signal to go to the ER in the first place. It's never going to bind SRP, so it's just going to be a protein that's translated normally in the cytosol, probably. If I had to take a guess. Again, let's talk about, let's talk about post-translational modifications. So, PTMs, talked about this already. DNA, transcription, great. Translation, great. Now, after proteins, we can do PTMs, post-translational modifications. You guys have already seen post-translational modifications. Guess what? Phosphorylations are post-translational modifications. There you go. That's one of the most common post-translational modifications you'll ever see. It's one of the most common types of post-translational modifications asked about on the MCAT. And what you see on the MCAT a lot is mimics. And I'll talk about mimics in a second, because they actually show up here. So what do they do? They give new properties to proteins. So like basically what you're doing is you're adding functional groups. So you add lipids, maybe you add a carbohydrate. Maybe that changes how the protein behaves. Stuff like that. You guys remember lipid rafts, right? Some of those proteins were anchored like GPI anchors, right? Some of them are, anchored by lipids. So maybe you have a lipid attached to a protein, and that leads to like this GPI anchored thing, and so it's a protein attached to the membrane, right? Things like that. So they can be used to confer new properties. They can be used to localize proteins. So some post-translational modifications tell proteins where to go. And then they can, of course, modify the activity of a protein. So like phosphorylation in this case can act as an on and off switch. So, yeah. Like I said before, yeah. So confer new properties. Basically, it can make it like lipid soluble, water soluble, stuff like that. Localize proteins, tells us where to go. And then modify the activity of the protein, phosphorylation, stuff like that. I'm not going to go through all of this. Basically, this is just another type of post-translational modifications. Insulin actually goes through a post-translational modification, if I'm not mistaken. Insulin is actually transcribed and translated as proinsulin, and then it's actually cleaved into insulin. It doesn't start as insulin. So that's actually something that you can remember. So like, oh, insulin's actually a product of a cleavage post-translational modification. So, okay, well, guys, I sure hope you know what this is at this point. What enzymes catalyze phosphorylation? Kinases. Which ones catalyze dephosphorylation? Phosphatases. Yeah. Yeah, so, what is this enzyme? That's a kinase. What is this enzyme? It's a phosphatase. And remember, phosphorylations can only occur at these three amino acids: serine, threonine, tyrosine. Why? Because you need to have an OH group. Remember, this O acts as a nucleophile for when you attack this phosphate. There must be a O, or it does not work. Right? You can't do phosphorylation on an asparagine. You can't do it on an arginine. You can't do any of that. It's got to be serine, threonine, tyrosine. These are very important. Very common question on the MCAT. Like, oh, where does phosphorylation occur? Oh, in this protein, where does phosphorylation occur? So yeah. Phosphorylation of a protein can either increase or decrease protein's activities. You guys have seen this. Pyruvate kinase, or not pyruvate kinase, pyruvate dehydrogenase, right? It's active when dephosphorylated and inactive when phosphorylated, right? What's an example? Another example, glucose 6-phosphatase, right? That's, I actually can't remember if that's one that's changed by that. But you get the idea, right? So pyruvate dehydrogenase is a great example of something that's edited by phosphorylation or modified by phosphorylation. Pyruvate dehydrogenase is active when dephosphorylated and inactive when phosphorylated. I think glycogen phosphorylase, actually, I think, is the same way. Glycogen phosphorylase, remember, is active when phosphorylated and inactive when dephosphorylated, if I'm not mistaken. So there are a whole bunch of enzymes that go through this process of being active or inactive based off of, is it a, they're active or inactive based off the phosphorylation state. So. Okay, yeah, how would you create a phosphomimetic mutant? Okay, so a phosphomimetic mutant means it mimics a phosphate group. Well, how do you mimic a phosphate group? Well, what is a characteristic of a phosphate group? Negative charges. Which amino acids have negative charges? Glutamate, aspartate. That's it. Replace serine, threonine, tyrosine with glutamate or aspartate, and there you go. You got a phosphomimetic mutant. Very common MCAT question. How? The researchers want to create a mutant that is always active. This protein is active when it's phosphorylated. How would they do this? Oh, they would do this by introducing glutamate or aspartate. Sometimes the protein will actually be completely different. Maybe sometimes the protein is like, the researchers want to introduce a protein that is never active. How would you do that? Oh, well, you would replace the threonine, serine, tyrosine with just a glycine or something. A glycine, or I don't know, what's a similar amino acid to serine, threonine, or tyrosine that wouldn't break it? Maybe a phenylalanine or something like that, right? Basically, you're just trying to put a similar amino acid that won't allow it to be phosphorylated in that case. So there's many different ways they can ask these kinds of questions. Oh, perfect. How would you create a phospho-dead or phospho-null mutant? So a mutant that doesn't work. They replaced these with alanines, right? So it can't be phosphorylated, it doesn't work. Okay, yeah, so in this case, I think the main thing to keep in mind, I don't think you have to memorize these, but they do say to add it to your note card, so I guess if you want to memorize them, you can. I would just add it to your note card. The main thing to keep in mind here is that if you see a serine or a threonine followed by a hydrophobic amino acid, it's going to be protein kinase A. If you see a serine and a threonine just on its own, I think X is supposed to be any amino acid. Actually, yeah, now that I think about it, this actually needs to be very specific. So if you see double arginine followed by any amino acid with a serine or threonine followed by any hydrophobic residue, it's got to be pKa. If you see proline, any amino acid, serine, threonine, proline again, it's going to be MAP kinase. If you see these guys here followed by a tyrosine with any amino acid, and then proline, and then phenylalanine, it's going to be ABL. So these, I would just write these down, honestly. These is too much to memorize, too much of a pain. So other kinds of post-translational modifications, ubiquitination. You guys talked about ubiquitination, actually, in 441. So abundantly used post-translational modifications used to, signal deletion and other things. So ubiquitin is very small protein, 76 amino acids. Attaches to lysines. It forms like an amide bond, basically. So ubiquitin itself has 7 lysines. And basically what happens is if you bind one ubiquitin, you can use that ubiquitin as a branch point to add more ubiquitins. So ubiquitin, 7 lysines. Each of these can be served as conjugation points for another ubiquitin. And so this, there are many different patterns of ubiquitination, and so each of them has their own different meaning, basically. Again, I am not going to go over all of this. I do not think this is important. Again, do not memorize this. Just know that ubiquitination can direct proteins to many different fates. There you go. Very important to keep in mind. Funny enough, this is just going through E1, E2, E3 again, which you guys already did in 441. I'm going to skip all the way down to this slide. So here we're going to have this. So remember, E1 is just the ubiquitin activating enzyme. It basically takes ubiquitin, activates it, and then attaches it to E1. E1 then binds to E2, which is ubiquitin conjugating enzyme, and then E1 basically transfers the ubiquitin to E2. And then E2 binds to E3, and E3 binds to specific target proteins. So these all complex together, and when E2, E3 plus this protein are bound together, E2 transfers its transfers its ubiquitin onto the protein of interest. And, this E3 is called ubiquitin ligase. And so from there, you can basically target this protein with polyubiquitin chains, do all sorts of random stuff with it. So, yeah. Yeah, you guys have already seen this before, so I'm not going to really talk about that too much. Okay, the one thing to keep in mind is actually, I talked about this in my 441 video, but there are very few E1s, decent amount of E2s, but there are so many E3s. And in my 441 video, I made it clear why there's so many more E3s. Because remember, E3s are protein specific. They have to be specific for the protein that they want to ubiquitinate. Otherwise, they're going to cause all sorts of problems, because they're going to start... That means that if the E3s are general, then they're just going to ubiquitinate everything, and then everything's going to get ubiquitinated. A lot of things might get deleted. They're going to get put in places they're not supposed to be at. So all sorts of problems with that if you just ubiquitinate everything. Also, a couple other things with post-translational modifications. So coenzymes and cofactors are actually other examples of post-translational modifications. So hemoglobin, the heme in hemoglobin is a post-translational modification. Retinol and rhodopsin is a post-translational modification. These are things that are very important in proteins. So the heme is very important in the protein of hemoglobin because it helps store the oxygen. The retinal is very important in rhodopsin because it's what allows rhodopsin to actually change conformation when light hits. There's also things like myristoylation, prenylation, and farnesylation. These are different types of lipid additions. So I would actually copy these down. I think I got tested on this, so worth having on your note card. So basically what you're doing is you're taking a protein and you're attaching a hydrophobic lipid to it. That's all it is, though. So this right here is a myristoyl anchor, this is a palmitoyl anchor, and this is a farnesyl anchor. So, yep. So myristoylation is this one, prenylation is this one, and then farnesylation is this one. Oh yeah, and the locations that they occur at is different. So for myristoylation, it's going to be at the N-terminal glycine. For prenylation, it's going to be at a C-terminal or internal cysteine. And then for farnesylation, it's going to be at a C-terminal cysteine. So there's a couple of things to remember for this. Oh yeah. These are just actually some examples of proteins that use these different anchors. So ARF uses myristoylation, RAB uses prenylation, and RAS uses farnesylation. So don't know if these are too important to know for now, but these are things that you should definitely keep in mind. But other than that, it's actually the end of this video. I do hope it was helpful for you. A very quick one, actually. It's like 20 minutes. Yeah, other than that, I hope this is a helpful video. I know it's kind of quick, but I don't want to take too much time on these because I know that, you guys have other classes and these lectures are dense, and I want to try and make sure I'm condensing the hard information down into what you really need to understand for these exams. So, yeah. Other than that, though, hope you guys have a good day and happy studying.